Royal actin makes queen bees have a different caste by Lauren Justice. Apis mellifera is the honeybees that we all know and love. Mellifera tend to be large in individual size as well as colony size. They use thermoregulation and are able to survive in the winter better than other species of bees. They are studied because they are one of the most important pollinators in the United States. Queen honeybees, so Queen Apis mellifera, are fed royal jelly when they are larvae. Queen bees live longer than any other kind of honeybee, and they are also the main reproductive figure in each beehive. Some components of royal jelly are royalism, royal actin, and gellins. So royalism is an antimicrobial property, also known as an AMP, and they can be used as a defense system against bacteria. Royal actin is one of the most important components of royal jelly, and it's the main cause of why there is a caste differentiation. And it's also the component that we'll be focused on today. And gellins are also antimicrobial property, and they are mainly used as a way to fight against bacteria. So, how does royal actin and royal jelly lead to caste differentiation? So, royal actin induces queen differentiation in honeybees. In this study, the researcher decided to test different components and temperature of royal jelly. They tested normal royal jelly as the control group, and then they tested royal jelly at 40 degrees Celsius for 30 days, used casein 450 kDa, royal actin, and E-royal actin as the variants. From this study, they found that the royal actin was the component that produced the most queen-like features. In the figure to the left, it shows that the royal actin had the highest ovary size and weight at adult emergence, but it had the lowest developmental time. Those are all characteristics of queen bees. And you can even see in the pictures right there, the difference in size between royal actin and the other variants. So differences in the morphology, physiology, and gene expression of honeybee queens and workers reared in vitro versus in situ. So in this study, the researchers decided to test if the natural diet of honeybee is actually different than a modified diet for in vitro honeybees. So they kept track of the differences by looking at the external characteristics of the worker bee and queen bee. The modified diet did not have royal actin in it, and differences were found between the comparable types. In the figure to the left, you can see that the body mass and spermatheca size were larger in the in vitro honeybee queens than the control honeybee queens, but the ovary sizes were larger in the control group. As the queen bee is the main reproductive figure in the colonies, we can assume that one of the reasons why the modified diet did not produce the hoped outcome is because the modified diet did not have royal actin. So apology, royal secretes in the queen's fat body. So in this study, the researchers discuss how royal actin goes through the epidermal growth factor receptor pathway, also known as the EGFR pathway. So the royal actin activates the EGFR pathway. This pathway is not activated in worker bees, but it is in queen bees, which is another main difference between the different castes. The EGFR pathway starts to release different secreted factors than those secreted in worker bees. In the figure to the left, it shows the different secreted factors that are released in the EGFR pathway. And if you look at the figure, you can see that it has systematic growth that is secreted, developmental time is also secreted, lifespan extension, and ovary development. So in conclusion, we can assume that the component of royal jelly, royal actin, has great influence on caste differentiation. Royal actin helps to increase the size of ovaries and weight at adult emergence, but it also has been shown to decrease the amount of development time. These are all important features of a queen bee and are the differentiating factors between queen bees and worker bees. So, the significance of this question is because the colony depends on the queen to survive. The queen is the main reproductive figure in the colony, so without a queen, the colony won't survive. So it is important to know how the components of royal jelly affect the larvae to ensure that there will be new generations of queen bees. Not only is it important for the colonies as a whole, but it's also important for us as humans because honeybees help pollinate all the plants around us and can help promote healthy lifestyles for us. And here are my references.